All right, we're going to conclude our study of the cell cycle by a look at what regulates the cell cycle. It doesn't just go round and round and round with nothing controlling it. That's what we call cancer. So what we're going to look at is what controls it and how we know what controls it. So here's our first experiment. And the first thing you need to understand is that the arrows represent time. This isn't uh, five separate dishes. This is the same dish over time. And in this first dish, we have a cell culture. Whenever you're growing cells in a petri dish with nutrient media, that would be called a cell culture. So it's a cell culture of fibroblast cells, which are some of the most abundant cells in your body, probably the most abundant kind of cell or cell type in your body, because they make up connective tissue, which is, really the bulk of your body. Um, so we've got these fibroblast cells growing in a petri dish with liquid nutrient media, and they multiply, they divide. And as they divide, they come in contact with each other and in contact with the dish surrounding them, or the edge of the dish, you could say. And then they stop dividing. And they stop, and they don't continue to divide and pile up on top of each other. They form one layer and stop. And so the scientists that were performing this experiment were kind of wondering why that happened. Why is it that the cells don't continue to divide and divide and divide? What stopped them from dividing? Well, it turns out that they identified what they call density dependent inhibition. Density meaning the density of cells, how many cells there are. And the more cells there are, eventually there are so many cells that they're all in contact with each other. They're all and they're in contact with the edge of the dish. And all that contact, when a cell is completely surrounded by something else, other cells or the edge of the dish, they stop dividing. And they also identified, in relation to why the cells didn't pile up on top of each other. Turns out they need to attach to a non-cellular substrate. And that's what they refer to as anchorage dependence. They need to be anchored to an extracellular substrate and a non-cellular substrate, so they can't pile up on top of each other because then they would be attached to other cells. So again, these are referred to as density dependent inhibition. When there are so many cells that they're completely surrounded by other cells, and that's a signal to stop dividing. And if they're not attached to something, a substrate, they will not divide. And they demonstrated this and went on with the experiment by removing a swath of cells right down the middle of the dish to see what would happen. And lo and behold, because those cells were not in contact with other cells anymore, they started to divide. So a swath of cells is removed, and that represents an injury. So think of this as skin, and this would be a cut, right? And the cells on either side of the cut are no longer in contact with other cells. Those cells have been removed by the cut, or those cells have been separated by the cut, and so they start to divide. And that's what we're seeing here. The darker blue are representing the new cells that are dividing and filling in. You could also say healing the cut, healing the injury, healing or refilling the swath. And they will do that um, until they come in contact with each other. But by putting a swath down the middle of the dish, what was removed was density dependent inhibition. If you remove an inhibition, that allows for whatever is inhibited to happen. In this case, the cell cycle or my cell division was inhibited, and the inhibition was removed. The density dependent inhibition was removed, allowing the cells to divide. And they divided until they formed one layer and then stopped. And they stopped then for the same reason they stopped in the original or at the beginning of the experiment, because they were completely surrounded, they were density dependent inhibited, and they didn't pile up on top of each other because of anchorage dependency. So the cells continued to divide until they were again density inhibited, and they stopped. So what this suggested to these researchers is that the cell cycle can be turned on and off, and that there was something outside the cell that was signaling the cell to either start dividing or stop dividing. Um, and so they were referred to as extracellular regulators because they come from outside the cell. Regulators of the cell cycle, signals that come from outside the cell to tell the cell to either start dividing or stop dividing. Extracellular regulators. So these signals seem to be involved in regulating the progression of the cell cycle, in other words, stopping and starting it. So you need to know density-dependent inhibition and anchorage dependency as extracellular regulators of the cell cycle. Here we see another experiment that was done that shows that there are intracellular regulators, internal regulators of the cell cycle. And what's being done here is what's known as a micropipette. This is a very, very small glass pipette, so small that the tip is so fine that it can actually poke through the plasma membrane and not destroy the cell, not kill the cell. So things can be injected into cells and things can be drawn out of cells through these little micropipettes. And that's what's being done in this experiment. I'm very familiar with these because I did work with mouse eggs. I injected things into mouse eggs in my graduate school experience. But you'll identify this cell as being in metaphase. It's very clearly in metaphase. And what's being drawn out of the cell is a little bit of cytoplasm and everything that's dissolved in the cytoplasm. So by taking out this sample of cytoplasm, all the different molecules, all the different proteins, the enzymes, and possibly signal molecules that might be dissolved in the cytoplasm is being drawn up with it. So that's what's being done here. A little bit of cytoplasm being drawn up in a micropipette. Then in cell 2, so these are two separate cells, unlike the other, the previous diagram was the same dish over time. Here we have two separate cells. So cytoplasm is drawn up from cell 1, and it's being injected into cell 2. And hopefully you've identified that this cell is in G2 of interphase. You can tell it's in G2 because there are two centrosomes that results from the doubling of organelles that takes place during G2. So this cell is in G2, and the cytoplasm is being injected in. And remember, everything that's dissolved in the cytoplasm is going along with it. And what follows that is time. So we're looking at the same cell over time. So this also is cell G2 or cell 2 in G2. Well, it's no longer in G2. It's still cell 2, but it has progressed to begin mitosis. So it's gone from G2 immediately after injecting the cytoplasm from cell 1 into cell 2 here that was in G2, it enters into prophase of mitosis. So a description would be that the injected cell soon transitions from G2 of interphase to prophase of mitosis. So what this suggests, and what it suggested to the researchers, was that something in the cytoplasm, some cytoplasmic factor, activated mitosis. And this would be referred to as an internal regulator. It's inside the cell. It's an intracellular regulator that regulates the cell cycle and cell division. And once they discovered this, the search was on for what that thing was that was dissolved in the cytoplasm, because there are hundreds of thousands of different proteins and compounds that might be dissolved in the cytoplasm. So they looked and looked, and they finally discovered a cytoplasmic protein that acted as this intracellular regulator, and they named it cyclin, because not only did it control the cell cycle or was related to the cell cycle, it itself cycled. So that's how they made the connection. They saw that the concentration of this protein would increase and then decrease and increase and decrease along with the cell cycle. So what they finally concluded was that it was regulating the timing or the rate of the cell cycle, as opposed to the extracellular regulators, which either turn the cycle on or off, or start or stop the cycle. These internal regulators are regulating how fast the cycle goes, the timing or the rate of the cycle. So they concluded that what they drew out of cell 1 in this experiment was cyclin, and what they injected into cell 2 was cyclin. And that cyclin then triggered the onset of mitosis. So we know it's been discovered experimentally how cyclin is acting. So again, when they discovered cyclin, they realized that cyclin was increasing and decreasing its concentration. So that's what this graph is showing, the cyclin concentration over time, but in relation to the cell cycle. So when the concentration of cyclin is high, mitosis is taking place, and when the concentration of cyclin is low, the cell is in interphase. And then the cyclin concentration builds up, and the cell enters mitosis. And then the cyclin concentration goes down, and the cell goes back into interphase. So it's cycling. Just like the cell cycle is cycling, the concentration of cyclin is cycling. So it's acting as an internal regulator. It was concluded that because cyclin cycles along with the cell cycle, that it is somehow related and possibly regulating the cell cycle. And because it's inside the cell, it's an intracellular regulator. Further research discovered how cyclin was acting on the cell cycle, and it involved another protein called CDK, cyclin-dependent kinase. CDK stands for cyclin-dependent kinase. So this is a protein which its activity is dependent on cyclin. If cyclin's not around, it doesn't do anything. But if cyclin's around, then cyclin binds to it, and the combination of the two form a molecule that's known as MPF, which stands for mitosis promoting factor. So this combination of cyclin and CDK, cyclin and cyclin-dependent kinase, acts on the cell to cause it to enter mitosis. It causes it actually to cross over the G2 point. Right at the end of G2, in the cell cycle in interphase, there is a stopping point, a checkpoint, and the cell stops at that point and does not proceed into mitosis unless it receives a signal to do so. And in this case, it's the internal regulator cyclin, the intracellular regulator, that triggers that by binding to CDK, forming MPF, which promotes mitosis promoting factor. And that causes the cell to pass the G2 checkpoint and go on to start mitosis, go into prophase. And here's that explanation. The diagram here has to do with extracellular regulators, and really it has to do with all extracellular signaling. So this is a very general diagram that shows how extracellular signaling works, how the message from outside the cell is sent inside the cell. So here we have a signal molecule, and that signal molecule might be what we call a growth promoting factor, and that would trigger cell division. The way you are growing is through cell division. Your body, as it grows, is getting more and more and more cells. So growth promoting factors are going to trigger cell division, signal cell division to begin. In other words, start the cell cycle, or cause it to proceed to cell division. So the signal molecule is going to bind to a receptor on the plasma membrane of the cell. That receptor then communicates with a signal transduction pathway inside the cell. So these are proteins, because proteins do everything. The receptor is a protein. The signal molecule is typically a protein. And so the receptor is going to activate one of these proteins in the signal transduction pathway, which is going to activate the next protein in the pathway, which will activate another protein in the pathway. And these pathways can be very long and also branching to activate other proteins. But the bottom line is that eventually something inside the cell is going to be activated and turned on. Some activity of the cell is going to start. And so it might activate something that's in the cytoplasm, or it might turn on a gene in the nucleus. Either way, it's going to activate something within the cell. And here's the explanation of that. So this is the idea of anchorage dependency and density dependent inhibition, but also signal molecules. So signal molecules will bind to receptors And the idea, as far as anchorage dependency and density dependent inhibition, there are proteins in the plasma membrane that sense when a cell is next to another cell, or in contact with the edge of the petri dish, or in contact with an extracellular um, substrate. So all those are examples of extracellular regulation, either turning on or off the cell cycle. So you need to know anchorage dependency, density dependent inhibition, and signal molecules, growth promoting factors like growth hormone, for example, as examples of extracellular regulation. Intracellular regulation, you want to remember cycling as the example. Sorry about that pause. I was just giving you time to realize that this came up and that all this came up, and had time to copy it down along with this. Finally, then we're going to take a look at what causes cancer. And what cancer is a loss of cell cycle regulation. What leads to cancer, what causes cancer, is when the cell cycle just breaks through its regulation. Nothing can regulate it. The cell cycle continues on, and the cell divides and divides and divides and divides. And it turns out that what causes that to happen are mutations in genes. And not just one gene, but a number of genes have to be mutated in order to cause cancer, in order to eventually lead to cancer. And we call those genes that are potentially cancer causing genes, or genes that when they mutate cause cancer, we call those oncogenes. And you may be familiar with the term that refers to a doctor that is responsible for curing cancer or helping patients that have cancer, an oncologist, right? Oncogenes, oncologist. So this onco prefix has to do with cancer. And the genes that are oncogenes are any of those proteins that are in that signal transduction pathway we were looking at on the previous slide. Any one of those proteins, if it mutates and it's stuck on all the time, could cause the cell cycle to just keep running and running and running. There are things that might cause these mutations, and that's what we call a carcinogen. So a carcinogen is a mutagen that will possibly cause a mutation leading to cancer. So what we're looking at specifically in this diagram is the development of colon cancer, which is a very slow growing cancer, and it's recommended that people over 50 have a colonoscopy where they send a camera into the colon and look for growths like this, possible precancerous or cancerous growth. And if they find them, they can just snip them out. But it's important to do that every 5 years or so. And 5 years seems to be often enough because it is such a slow developing cancer. But what we're looking at here is the colon, the large intestine, the colon. Those terms are really one and the same. And these are polyps. So these are the stages of polyps that a doctor would want to catch, or an oncologist would want to catch early stages, and they would just snip them out. So along with the camera, they can send along tools like scissors, very small scissors, to be able to snip out any polyps that they find in the colon when they do the colonoscopy. So a polyp is a relatively small, uncontrolled, slow growth of tissue. It's not cancer, but it could be considered precancerous because if you let it go, it could develop into cancer. Tumors are relatively large. So polyps are small, tumors are larger, uncontrolled growth of tissue. And the polyps are benign. Tumors can be benign, and what benign refers to is that they're not really hurting anything. A benign tumor is not hurting anything. It's not spreading anywhere. It really hasn't grown to the point where it's damaging surrounding tissue. A malignant tumor, however, would be considered cancer. A malignant tumor is to the size where it is invading and damaging surrounding tissue. So that's what malignant refers to. So if you go to the doctor and the doctor has diagnosed you with some kind of growth, you want it to be benign. That's the best case scenario, because then they can usually remove it and it's not going to grow back. If it's malignant, if they can try to remove it, but if they can't get all of it, then if they leave any cells at all, it might grow back. It probably will grow back. And then the worst case scenario is being represented here by this arrow. And the whole reason that they show blood vessels coming in here, and these green things are representing the lymphatic system. So these are lymphatic vessels. Um, and what can happen is cells from a malignant tumor can enter the bloodstream or enter the lymph, which is the fluid that flows through the lymphatic system, and travel to other parts of the body and cause cancerous growths in other parts of the body. When that happens, that's the worst case scenario. It's called metastasis. So if a malignant tumor metastasizes, there are cells from that tumor traveling all over the body and causing tumorous growths or cancerous growths all over the body. That's definitely the worst case scenario. So metastasis is the spreading to other parts of the body through the circulatory or lymphatic system. So you may have heard situation if a woman has breast cancer, they'll not only do a mastectomy or a lumpectomy and take out the tumor, they'll also take nearby lymph nodes because it's possible that some of the cells from that malignant tumor may have traveled to lymph nodes. And that's not a very happy way to end our look at the cell cycle, but it does emphasize the importance of understanding the cell cycle.